Hello and good afternoon, everyone. Uh, Today we are going to talk about the most common bacteria, the Helicobacter pylori. You can see I have written this Helicobacter pylori in the italic form because whenever you write this, the genus and the species, you have to underline. Okay, this is the genus you have to underline here and pylori. Okay, from this name we can just imagine that this is a helico, means like helical. Helical means we know about the helical in physics. We are talking about the helical spring like light. So this helico means it's a spiral. This is spiral. So bacter from this you just remember is the bacilli. That means it is rod shaped. And pylori means like it loves to live in the pyloric part of the stomach. Okay, especially in the pyloric antrum. Okay, from the name itself we can guess some of the general characteristics of it. Now regarding the morphology, it is spiral, cup shaped. Okay, so it is a spiral or cup shaped rod. And regarding the gram stain, it is a gram negative. It is a gram negative bacteria. So regarding motility, it is motile. So motile means it is motile with the help of flagella. So what type of flagella? It is a lophotrichous flagella. So what is meant by lophotrichous flagella? It means having a tuft of flagella, having a group of flagella at one end, not at the both end. Okay, at one end. So this such type of flagella containing bacteria, it is lophotrichous. And next, it is microaerophilic bacteria. Micro means what? Less. Aerophilic means what? Oxygen. So it loves less oxygen. So that less oxygen means how much? That is 5 to 10% oxygen. Okay, for the growth. Now regarding the biochemical test, it is catalase and oxidase positive, and most important, it is the urease positive. Okay, why urease positive? Because it contains enzyme that is called urease enzyme. Okay, that has also bigger role in the virulence factor of this Helicobacter pylori. Now let's go into the virulence factors. You can see in the virulence factor number one is the flagella. I have already said what kind of flagella, lophotrichous flagella. So because of the flagella, what is happening is that I have already said this bacteria only loves the mucous layers of the stomach. Mucous layer of the stomach. So inside the mucous layer, it can swim. Okay, it can swim with the help of flagella. It can goes deep into the mucous layer so that it can be protected from the acid also, or with the help of flagella it can go deeper inside. Okay, deeper inside, so that causes the its virulence factor. Next is, we know about this lipopolysaccharides. So what is lipopolysaccharides? We know that. So lipo, in every gram negative bacteria, right, it contains lipopolysaccharides. So lipopolysaccharides are also called as the endotoxin. So what is happening in the lipopolysaccharides of this bacteria is that this lipopolysaccharide shows the that this, because of these lipopolysaccharide, this bacteria shows molecular mimicry. The chances of Helicobacter pylori infection is greater with the Lewis B blood group. So the blood group antigen present in the Lewis B blood group, that is Lewis B antigen, and the LPS looks like similar, and because of that our the antibody antibody insists of killing insists of finding this antigen LPS of the bacteria. It will it will also damage to our one blood group antigen. Okay, that is the Lewis blood group antigen. So that incidence is greatest in the Lewis blood group antigen. Now the regarding number third is the urease enzyme. So regarding the urease enzyme, so what is urease enzyme means? It is an enzyme, you can see urea age. So this is something breaking the urea. So we know about urea can be broken down into the ammonia plus CO2. Okay, now with the help of this enzyme, this bacteria break down the urea into ammonia and CO2. Ammonia, so ammonia is what? Basic in nature. So it will create a microenvironment surrounding it. Okay, it will create a microenvironment surrounding it. What type of environment? Microenvironment, that is alkaline microenvironment. So because of the presence of alkaline environment also, this bacteria tries to protect from the acid of the stomach. Now the most important the genes like the BAC and the CAG genes. So what is BAC? BAC means it is a vacuolating cytotoxin A. Vacuolating cytotoxin A. So vacuolating cytotoxin A, the name itself suggests that it helps in the formation of vacuoles in the mucosa of the stomach, right? So it forms the pores, it forms the pores in the mucosa layer and causes the death of the cell. Likewise, the CAG. So CAG, it is, what is CAG? Cytotoxin associated gene A. Okay, the cytotoxin associated gene, it is also called as the pathogenic island. Pathogenic island. So what does it directly do is that it helps in the what? For me, it helps in the one system, that is type 4 secretion system. Type 4 secretion system. And what is type 4 secretion system? That is, they are the pathways used by the microorganisms to transport any type of proteins, any type of DNA across the membrane. Here the membrane is of course the mucosa of the stomach. So this CAG, now with the help of this type 4 secretion system, what is there will be the injection of some effector molecules into the mucosa of the stomach, and there will be some changes in the gastric epithelial layer. That is, there will be the expression of the proto-oncogenes there, cytoskeletal rearrangement will be there, and similarly the activation of pro-inflammatory substances. So these will all cause the irritation to the mucosa, and finally the mucosa will be irritated and it will cause inflammation there. So this is causing the virulence factors of the Helicobacter pylori. Now adhesions, it helps to what? Adhere to the host cell. Okay, that is the stomach cell. So now the lab diagnosis. So regarding the lab diagnosis, it has the invasive test and the non-invasive tests. So regarding the invasive test, what we do is, one is the endoscopy guided biopsy. From the endoscopy, we take the biopsy, especially the antrum tissue, and that antrum tissue now will be go into the histology. So we will see the tissue type. Okay, so with the help of histology, if there is sudden change in that cell type, then we can also have the diagnosis. Next thing is that tissue can be stained. So that tissue can be stained. So what are the staining techniques? One can be done, of course the gram stain can be done, and next thing is one stain is there, that is called as what type of stain? That is called as silver stain. So what is the exact name of that silver stain? That is Warthin steri stain. Warthin, Warthin, Warthin steri stain. And next thing is, of course, we can also do the Giemsa stain also, right? Now we can also okay, or we can also take a broth. Okay, we can also take a broth. Broth means it is a liquid media containing the urea and a pH indicator, and we add the biopsy tissue. If there is change in the color of the pH indicator, then it also helps in the diagnosis. So how is this happening, the change in the color of the pH of the indicator? That is all done by the urease enzyme. So urease enzyme will convert the urea into ammonia, and thus this formed ammonia will change. The urea will change the color of the pH of the color of the indicator. So that is called as rapid urease test. Now the culture is not usually done, right? So but the media for this is Skyros media, because it is a microaerophilic. We have to maintain very less amount of oxygen, so we generally do not use practically the culture. So now non-invasive test, it is usually done in the hospital, that is the urea breath test. So what is in the urea breath test? That the patient is given a urea solution to drink, right? A patient is given a urea solution to drink. That urea solution is labeled with carbon isotope, C14 carbon isotope, and now after 15 minutes, now patient is now advised to take rest for some time, and after 15 minutes the patient blows into a breath packet like things, or you can say just like our tube. Okay, and blown into tube, and that is analyzed with the help of breath analyzer. And in the breath analyzer, we can see the amount. We can see the amount of that isotope, carbon isotope, and with the help of this technique also we can do the diagnosis of this bacteria. So the UTI bacteria is doing, so it is the most it is doing in most of the cases. So non-invasive test so it is also beneficial to the patient, so it is convenient, it is convenient also, and its price is like the 1800 rupees Nepali. And now next thing is serology. Serology, we can also detect antibodies like by the ELISA test. So this type of test is usually done for the epidemiological purpose, right? Now regarding the advantages of this bacteria is found to be protective against protective against the gastroesophageal reflux diseases. And even we know that the long-standing gastroesophageal reflux disease can also lead to the what? Barrett esophagus. Barrett esophagus is of course the most common cause of the adenocarcinoma of the esophagus. Okay, so these are, so and in some of the allergic disorders like asthma, so it is also found to be protective. Now disadvantages, of course, this is one of the major cause of the chronic gastritis. 90% gastritis is chronic gastritis due to the Helicobacter pylori infection, and this Helicobacter pylori infection is usually causing the what? Duodenal ulcer. Which type of ulcer? Duodenal ulcer, and of course the gastric ulcer also. Okay, so these are the disadvantages of this bacteria. Okay, so this bacteria is of the major concern in the developing countries. Okay, so thank you all. This is all about the Helicobacter pylori. Thank you all. Thank you.